Thank you for inviting me today to talk on what's going on in the United States Food and Drug Administration on organs on a chip. From the development program we started to hoping to fund and qualify for regulatory acceptance. So next slide. First, I want to say that FDA encourages the use of new testing methodologies. We want to work with regulators worldwide to incorporate new testing methodologies into regulatory standards if certain qualification criteria are met. And we also found that it's important to ensure our regulators' familiarity with techniques before they see them in a regulatory submission, so that when they see them for the first time in a regulatory submission, they understand the type of data they need. So any technology, including organs on a chip, to be considered for regulatory use has to be proven to be reliable, robust, reproducible, and fit for the context of use that it's proposed for. I want to mention that we do not fund the development of MPS, which is the same as organs on a chip, OOC, that you use for regulatory use in the support of new products. Let me talk a little bit about the history of FDA's involvement with organs on a chip, or microphysiological systems. In 2010, FDA and the National Institute of Health Common Fund awarded money to the Beast Institute for biologically inspired projects in Harvard University to develop a heart-lung micro machine, as it was called at the time. And that was our first introduction to organs on a chip. In 2011, DARPA, which is the Defense Acquisition and Research Agency of the Department of Defense, approached the FDA's Office of the Chief Scientist requesting that we work together to develop a human body on a chip for medical countermeasures. So they funded research and involved FDA from the very beginning of this program to help ensure that regulatory challenges for reviewing drug safety and efficacy were being considered during the development of this MPS platform. And later, NIH's National Center for Advancing Translational Science also funded part of this tissue program, and we have been a partner throughout this program until today. And I think what's critical here and what FDA is stressing is that it's important to have regulators at the table at the beginning if the aim of any new tool is to use it for regulatory use. For organs on a chip, at the beginning, we talked about what the regulatory uses could be, what questions we wanted to answer, what reference compounds we wanted, what context of use we want to see, and we continue today to talk to and work with our global partners, including all of you here, to develop this and other new predictive toxicology tools. In 2017, FDA announced its predictive toxicology roadmap. This is really not a roadmap, but more an overseeing of what we wanted to do as part of incorporating new predictive methods into regulatory use. The predictive toxicology roadmap listed what we were going to do. We were going to work with our global partners. We were going to train our regulatory agents to understand it. We were going to develop performance criteria, and we were going to talk as one agency on developing new tools. Next slide. This was a great beginning, but then we decided to bite off one part of new predictive toxicology tools and look at alternative methods. And we formed an alternative methods working group under the Office of the Chief Scientist and the Office of the Commissioner with members from all parts of FDA, and this is a committee that I chair. And we want to discuss alternative activities across FDA for use in toxicity and efficacy testing. We wanted to interact with our U.S. and global partners, such as all of you here, to facilitate discussion, development, and acceptance of regulatory performance criteria for such assays. And we wanted to update all of our stakeholders in real time. So we developed a website, and it's listed here, or you could just type into Google FDA alternatives to tell our stakeholders what we were doing in real time. And we also have an email address, alternatives@fda.hhs.gov. which we are encouraging all stakeholders to talk to us about issues. What are the objectives of this alternative working group? I discussed a little bit about it, to look at new in vitro, in vivo, in silico methods for research, training, and communication, to interact with our U.S. federal partners and global stakeholders, such as all of you here, to facilitate discussions such as this one on the development of draft performance criteria for assays. We wanted to establish a dialogue and develop partnerships, explore regulatory science applications for new technologies, to identify performance criteria for new technologies, including microphysiological systems, by engaging FDA experts and FDA stakeholders throughout multiple public-private partnerships, which are partnerships between the government and industry and NGOs and other people. All of us were getting emails that people had new technologies that they had developed, especially microphysiological systems, and they wanted to preview them to FDA. So we have developed a webinar series on alternative methods. This is an opportunity for developers, both academic or industry, to present new methods and methodologies to FDA. We hold the webinars monthly. We advertise them to all FDA scientists, and if selected, developers it would not constitute that we're endorsing or qualifying that method, but we would help if people at FDA we would help some of our centers if they want to get involved with these technologies early on. So it's a way of us looking at new things, but also another way of encouraging dialogue. And this is all on our website, but next slide. And these are the criteria for being considered for selection. FDA would like to be able to help people as they're beginning to develop new methodologies, but we just don't have the resources to do that. So we want to look at mature technologies which have a context of use. You've already developed what kind of questions or health standards, something that you could use your method for, including any publications that you might have. If you're using cells, talk about the cells, are they animal or human? As I said, the context of use, description of any gaps that you want. So we really want to know a little bit about your method. Is it mature? We want you to tell us what you think that this method might be used for. And we will advertise it to all of FDA, including what we have is our user group, which is a group of about 45 FDA scientists, research scientists that actually have organs on a chip in some of the FDA laboratories. So also on our Advancing Alternative Methods page, you will see some of our publications on alternatives, and they're linked so that you can get those publications. We may also have presentations that we've given, PDFs of presentations. We have one right now on organs on a chip from our acting commissioner, Dr. Woodcock, for people to look at. This is all public. And we will constantly update this with anything that will let people know what we're doing on all of these areas. Next slide, please. We also just published our first alternative report. You can see it peeking out there. This is learning how FDA is advancing new alternative methods, and it really gives you a snapshot of what we're doing in each of our program centers, including on organs on a chip. So FDA has 6 program centers, which would be foods, veterinary pharmaceuticals, human pharmaceuticals, biologics, which include the vaccines, devices, and tobacco. And so this is a snapshot of what we're doing on there and our big lab in Arkansas. And you can get that at our website and look at what we're doing. It has an introduction by our former commissioner, Dr. Hahn, our chief scientist, to really tell people that we are encouraging advancing more alternative methods and the 3 R's. So what I said before, the Alternative Methods Work Group, or the AMWG's first case, is looking at in vitro microphysiological systems, or what you're calling organs on a chip. We have because we were so intricately involved in the very beginning of this technology in 2011 when it was first started, and the DARPA grants went to the V's at Harvard and and MIT, and those were the two DARPA grants, and then there were several grants that went from NCAS to other academic people developing chips. So we wanted to define what FDA meant by microphysiological systems, so that when we talk about microphysiological systems and organs on a chip, this is what FDA meant. We wanted to develop partnerships. We wanted to develop performance standards. And we wanted to develop some sort of mechanism to request information from MPS developers and end users. And some of that is being done with our partnership, what's called the IQ Consortium MPS affiliate. The IQ is Innovation and Quality Consortium, MPS, Microphysiological Systems affiliate. Those are the farm people from a lot of the pharmaceutical companies that have formed. They have a IQ consortium and they form this affiliate to look at MPS. And we meet with them regularly and talk to them about what we're doing and what they're doing. Next slide, please. This is our draft definition for microphysiological systems. It's on our website. We wanted microphysiological system to include not only organs on a chip, but organoids. This is our draft working definition. You're certainly welcome to give us feedback on it. I won't read the whole definition because you'll probably see the slides, but once again, at alternatives@fda.hhs.gov, that's also the website that you use if you want to request coming to the FDA to give a seminar or request anything else of FDA, and that goes to my email address, other people's email addresses on the committee, and we try to be prompt. It's been a little bit hard with the COVID right now, but we're trying to be as prompt as we can because we realize that all of your feedback is important to FDA as we work together to develop new technologies. Next slide, please. So we encourage stakeholders dialogue, and you're all stakeholders, what we call stakeholders. We encourage you to talk to us about the potential uses of MPS, other predictive methodologies, and for both toxicity and efficacy. And the venues are what we said before, the AMWG webinars, meetings such as we just had a meeting on with the National Academy of Science and ILAR on microphysiological systems, bridging health and animal and human research. If you didn't see it, I will send you guys a link to all of the talks are on YouTube, and I'll send you a link after that. It was a really wonderful symposium that was done a couple weeks ago. We have other joint meetings on MPS. You can email us if you're interested in devices or drugs. There are pre-IND meetings where you come in and you talk to people about it. We have what's called a critical path innovation meetings, which you can come in and talk to the FDA outside of our regulatory application. The critical path is the path from that CDER calls the critical path. It's the path from discovery to approval is a critical path for pharmaceuticals. So there's a lot of different ways to communicate to us. Certainly, I have my email at the end of this too. And again, I encourage people to talk to us about it. We all want the same thing. We all want new predictive tools to get safe and effective products on the market. Next slide, please. So one of the ways that we're doing this is we have to learn more about organs on a chip is that we have the user group. And as I said, the user group are a group of researchers doing research on organs on a chip at FDA. Here are some of the CRADAs. A CRADA is a cooperative research agreement and development agreement that we can make with an outside organization to partner together on technology. We have a lot of different other ways that we can partner on technology. And if people give seminars at FDA and parts of FDA are interested in talking further, we help them with these different partnership agreements. So we have the tissues chip, which is the German chip. We have we just signed a bigger agreement with Emulate to look at several chips there. We have some chips from CM Bio and from Seattle. And if you go to the next slide, I'll talk a little bit more about some of the chips research that we're doing. In our CEDAR lab, which is CEDAR is the Center for Drug Research and Development, we have two chips. We have the CN Bio liver chip. CN Bio is the commercial company that was developed by MIT. When DARPA gave several $1 million to both the V's at Harvard and to MIT, the agreement was that both of them would have spin-off companies, commercial companies, that then could develop these further. And the spin-off company for MIT was CN Bio. CN Bio we have their liver chip, and we're looking at cell death, metabolism, biomarkers, and gene expression. We also have engineered tissue heart chip that we got from San Francisco, looking at contractility, calcium cycling, length of contractions. And we also have a heart-liver system that we got from UC Berkeley, trying to combine these two systems together and to see what we get. So these are some of the research that we're doing in our drug lab. The next slide. In our CBER lab, which is the vaccine lab, we're looking at regenerative medicine cellular therapies. We're trying to replace or regenerate human cells, tissues, or organs to restore normal function. So this is what we're isolating cells, expanding them, developing a scaffold, seeding it, and look at therapeutic tissue engineered products. Okay. And I don't believe these products are transferred into patients. They may be starting early clinical trials. So next slide, please. And we've also given an extramural grant that's outside of FDA by our Office of Counterterrorism and Emerging Threats. This is our medical countermeasure group. We awarded a contract to the University of Liverpool to understand the coronavirus diseases through analysis of non-clinical and clinical samples. This is in London, Liverpool. The project includes developing in vitro coronavirus models to inform medical countermeasures, and led by the Public Health England with a focus on the lung chip. So we're trying to not only look at what's going on with COVID, but try to be proactive and see if we can look at new models before we get the next pandemic, or in preparation for a possible next pandemic. So next slide, please. We also have an intramural grant that we are given to Tony Wang in our CBER lab to look at the emulate lung chip and study COVID with multiple strains of the SARS virus. We do have a BL3 lab in there, and he's studying antibody response in the lung chip generated by human plasma samples with high titers of antibodies. This is part of a CRADA that we just did with Emulate, and I think I'm describing the other two products. We're looking at the lung chip, their Emulate lung chip for COVID. We're looking at the gut chip in our vet drug lab for microbiome and antimicrobial susceptibility. We're looking in our drug lab, we're looking at the liver to compare it to, or we're just getting these chips to compare it to what we saw with other liver chips in our National Center for Toxicological Research. We're getting the blood-brain chip and the liver chip also to look at the blood-brain to look at possible Alzheimer's issues, and the liver chip to follow up on other drug metabolism. So next slide, please. And this is what we're doing at nctr. We have the tissue chip, as I said. We're partnering with Tissues, which is the German chip, to develop an MPS containing organoids for two tissues put together. And we want to look at rat to human extrapolation, to look at, we're looking at hepatic drug metabolism and spermatogenesis, and try to get some criteria for qualifying these chips. And as I said, nctr is just getting the two emulate chips from the liver and the blood-brain barrier. It's actually being delivered this week. Next slide. And CDM is our veterinary drug center for veterinary medicine. They are getting the gut chip that's been installed. They want to develop the gut on a chip model for determining the impact of antimicrobial drug residues on the human intestinal microbiome, including the development of antimicrobial resistance. This is for drugs used in food animals, and the drugs used in food animals contribute to antimicrobial resistance. And so they're trying to develop, their long-term goal is to develop standards for qualification of this model to look at antimicrobial drug products intended for use in food animals. So this is, as you know, is a very big issue. It's an ongoing issue for a long time, and they want to see if the gut on a chip, which is from Emulate, this is the Emulate gut on a chip, can help us with some of these answers. The next slide, please. So that was a sort of a roundup of some of the chips that we have. We also have a chip from South Korea, the Curios chip in our Seeger lab, which I probably should have described that work is going on there. And we have several organoids in our Seeger lab by Dr. Sani. That is, she's doing recent organoids, and she's actually the person that developed the first draft of our definitions for microphysiological systems and organs on a chip. So, as we said before, we've come away from what used to be the all-encompassing validation, where it's for everything, every endpoint, to what we call context-abuse qualification. You develop the computation. A context-abuse is a clearly articulated description delineating the manner and purpose of use for a particular approach or element or new tool. So you develop your context of use and you qualify it for that use using a model, an assay, or something. And if it's qualified, if it ends up being qualified, it could be used by you or by anyone in a regulatory application without putting any underlying data to show its reliance, relevance, sensitivity, specificity, etc. There's two. You could use in any of our applications. You could include a new method that we haven't seen before, and you can talk to the regulators about what kind of data you need to qualify for a use. It doesn't have to go through a formal qualification program, and we have one of those at our drug lab and also our device lab. You only go through the big formal one if you want to let the whole world actually use your method. So it's qualified, the analysis qualified for this, and anybody can bring them in. But you can come in, and we encourage you to come in to bring your new methods in on a pre-IND, pre-IDE, pre-BLA meeting with FDA with new tools to talk about how it could fit into your particular regulatory application. And that stays confidential. That stays in your application. And we don't share it with anyone, or the fact that your application is there unless you want to share it. So next slide, please. So, as I said, you start with a regulatory question or the context of use. What needs to be answered and why does it need to be answered? And that question determines how much qualification is needed for a particular assay. If it's a discovery or screening assay, you're not going to need much. We can look at a lot more uncertainty on the discovery one. Then if it's a pivotal study that might replace some traditional safety study. So it really, in my mind, is what is the consequences of getting a wrong answer? If it's a screen, you might not develop a compound that you would have developed ordinarily. If it's a pivotal study, you might put something on the market that really isn't safe. So, and that really defines how much performance, how much data we need about that in order to, and how much uncertainty we're willing to take on that data to use in a regulatory application. So you help define the applicable domain and limitations. So the domain is what kinds of criteria, what kind of compounds can your method use? That's the domain. And what are the limitations of your method? And you can come in with one context of use and then expand it over time. It doesn't stop you from using a method or something other than that context of use, but it's just that you have to put in a lot of compounds. I think a lot of the data to show that the extra uses are reliable, reproducible, etc. And I think it's very strongly that the reference compound used, the test compounds, positive and negative controls, should be determined by the context of use. I know that people are developing, and I meant use, not us, but us, we're involved in that too. So we should, sometimes people have reference libraries, but really, and you just say, oh, this is good for this or this or this. But I think you really need to know, what am I trying to show this method can predict? And so you want to make sure your reference compounds, your positives and negatives, really can predict the same thing that you're trying to predict with the new method. So next slide. And some other questions as you move towards regulatory use that you should think about. You need to have enough data in order to answer the question that you want to answer. You have to make sure the assay is mature enough. You have a stable platform. Are the cells, what about the cells? Have you done a lot to make sure the cell, have you followed good cell culture practice? Have you made sure that the cells are stable, or they are what they are, and that they stay viable for the period of time? What endpoints are you measuring? Are they predictive of in vivo events? How are you going to translate to humans? Do you want to use animal concordance data? Do you want to use human in vivo data? That's something that we're discussing now. What do you benchmark for these new methods for microphysiological systems? Do you have to have animal tips, or can you benchmark forward to humans? And that's something we're discussing, and we would like your opinion on it. Then what test compounds do you do? Do you need those compounds that have in vivo data? You need positives and negatives. As I said, you define the compounds the assay can assess and not assess. And then you need to say, what's the sensitivity and specificity for your method? So as you move towards regulatory use for any new methods that you have, including organs on the chip, these are the kind of questions that you need to think about as you're developing it. And next slide. So this is a little slide. I hope I gave you an overview of what's going on in the United States and at the Food and Drug Administration on organs on a chip. It takes time to get these new technologies. They are really exciting. Believe me, they are my favorite compounds. I helped start the program. I love organs on a chip. They're not standalone. They're not standalone tests right now. They're part of some sort of regulatory IATA in my mind to answer some questions, but they're certainly not ready yet to replace clinical trials with humans. But at some point we may get there. It will take time, but if we all work together, the FDA with all of our stakeholders like you, we can get them together. And with that, and the next slide just has my contact information. And thank you very much for inviting me here today. And I hope I hear from you. Thank you.